we want to use this webinar to showcase not only the modeling and simulation capabilities at ASIP, but also to give people at ASIP and university partners an opportunity to learn more about this topic. So today, my colleague Margit Winkler, also a senior scientist at ASIP, has offered to organize this webinar and has invited two very exciting speakers. But before I hand over to Margit, I wanted to give a little bit of info on how it is structured for those that are new. So we have two presenters today. Each one has 20 minutes, more or less, to present. And then we have 10 minutes of Q&A, and afterwards we will switch to the other speaker. So as you can see, the meeting is being recorded. This is why you can either ask the questions by raising your hand in the chat or by using the Q&A tool at the bottom. And this you can also ask questions anonymously if you don't want to be identified or on the video, on the recording. So with that, I want to hand over to Margit, who will moderate the rest of the webinar.
Thank you very much, Nico. I'm excited to do this for the first time. And even more excited makes me the fact that Svetlana Findrik-Blažević has kindly agreed to give us an idea about her modeling activities. So while she is setting up her slides, I will give a short background about her. So Svetlana is active at the Department of Reaction Engineering and Catalysis. She became assistant professor already in 2009 and associate professor in 2012. And for 2 years now, she is full professor at the Department of Reaction Engineering and Catalysis at the University of Zagreb. She holds really numerous national awards, and she's also exceptionally successful in the acquisition of EU-funded projects. So I just want to highlight maybe one of them, and this one is called the CirculH2 project, which deals with hydrogenase enzymes for the large-scale deployment of hydrogen, so hydrogen in gaseous form as an energy carrier in industrial biotechnology. But without taking more of your time, Svetlana, please go ahead and talk to us about the application of kinetic modeling in the development and optimization of biotransformations. Thank you.
Thank you very much. Thank you, Margit, for your kind invitation. It's I'm very happy to be here. Considering bad internet connection, I will shut down my video. So I hope this will not, you will not mind that. Margit said, I am dealing with topics related to kinetic modeling. And in my group, we are developing kinetic models and use them for optimization of different biotransformations. So in the first with you, sorry, just a second. I will share with you aims of the studies we carried out in my lab. So by using reaction engineering approach, want to understand the process. So this means we want to understand what is going on in the reaction mixture in the reactor. We want to find which are the bottlenecks of different reactions. And I'm not talking about solely of the cascade reactions, but also of the reactions which we usually call simple ones because they are just one equation, two substrates, one product, or something like that. Solutions for. And by using this approach, by developing these equations, kinetic models, we want to perform a reaction and reactor optimization, which means that if we consider that in biocatalysis, we have many inhibitions by the substrate,
I'm afraid the internet connection seems to be down. At least I cannot hear much at the moment.
Yeah, I also cannot hear Sustana anymore. Okay, maybe.
Okay, you don't hear me at all?
Okay, now you are back.
You were gone for a minute or
I'm
so.
back.
Yeah, so we didn't.
So you haven't heard anything I spoke about?
Unfortunately, we didn't hear much on this intro slide. Yeah.
We heard about half of the aims, but then you got lost.
Okay. I can then, let's say, try to repeat, but please let me know if you don't hear me, so I'm aware of this. So we want to perform reaction and reactor optimization. This means that we want to find the best possible reaction conditions for the reaction and the best potential reactor, because considering all the inhibitions that are usually present in the biocatalytic systems, reactor choice is very important. So when I say optimization, we want to obtain maximum possible process metrics. This is something I will talk about a bit later in more detail. And we want to evaluate the viability of the process. This means that we test the economic viability of the process. So the approaches we use are mostly reaction engineering, which is, I'd say, giving us the most information, whereas in certain cases where we cannot handle the system, where we don't have the chemicals we need for experiments, then we use statistical modeling as well, or we use the stochastic approach. So in this talk, I will give you some examples. First of all is the example we have been working on in the past, which is the synthesis of R-hydroxy gamma butyrolactone. And the second one is an example of the project Circle H2, which we started working on like a year and maybe a year and a half ago or a bit less. And of course, this is not finished yet. So I will start by saying that reaction engineering is a scientific discipline that has the aim and the purpose to industrially produce a compound in a reactor. So how does it work? It works by investigation of reaction kinetics, and this means carrying out a multiple of experiments where we get such series of data, and then we formulate the equations which describe the reaction system, and then we can use this model to predict what will happen if we change the conditions, reaction conditions, or if we change the reactor setup. So typical goals of process optimization are the ones that are presented here on this slide. So usually those that are also for industrial production, of course, like volume productivity, product purity, product concentration, substrate conversion. So we can select more than one, of course, which makes optimization a bit more complex. However, it works quite well. So in the meantime, when we are talking about optimization, we can also talk about minimization of the consumption of chemicals and cofactors. So this is something that can also be optimized. So I will share with you the methodology that we use in our lab, which we sort of developed throughout the years, which is based on selecting proper reaction conditions for each enzyme or each reaction system. So if we have a single biocatalyst in the reaction mixture, then it is easier, of course. But if we have several biocatalysts, like in the cascade reaction, then we have to choose the conditions which are good for both or two or three enzymes. So sometimes this means that certain enzymes cannot really work together, and then you select a cascade reaction with one part, several steps that are not conducted simultaneously. But let's say that we select the reaction conditions and then we perform the kinetic studies, which are mostly performing the reactions at its initial stage. Initial reaction rate method is often used. And from the collected data, we can estimate the kinetic parameters, which are maximum reaction rate, Michaelis constant, inhibition constants, and we use nonlinear regression analysis. So once we have this set of parameters and we have our reaction scheme, we can suggest the first iteration of the model, which can then be combined with mass balance equations for a certain reactor. And then we can use these equations for model validation. Of course, model validation means that we need to perform more experiments. This means following all reaction components in the reaction mixture during time until the reaction is done. And then we see if the model fits the data. If not, in many cases this is the case, then we have to perform the operational stability studies for biocatalysts, and then we can evaluate and quantify how stable is our biocatalyst. When we have this data, then we can introduce them to the model as well. And usually then we can use the model. The model describes the data well, and we perform model simulations to find the best conditions for the reaction and to choose the best reactor mode. Now, once we have done that, we can also use these optimal reaction points to test if those are really working that well as with simulation. And we can also use these optimum points to evaluate economic viability of the process, which I will say something about later on as well. So the first system I'd like to show you how this works is this cascade here, which is carried out in 3 steps. The first 2 catalyzed by halohydrin dehalogenase, and the third one catalyzed by nitrilase. So starting from this compound here, 1,3-dichloro-2-propanol through epichlorohydrin to this hydroxynitrile, and towards this compound, which is pharmaceutical building blocks for different interesting pharmaceuticals. So when we set up a cascade like this, we are interested in answering several questions, such as what is the best way to perform this synthesis? Are there side reactions that we need to take into account? What reactor we should use to find the best performance? And what will be the reaction conditions? So in order to do that, as I said previously, we perform a lot of experiments. So in this case, and I will not go into details here because of the time constraints, we select each of these steps and investigate them separately. And for this first step, in epichlorohydrin synthesis, we found that this reaction, it is of course an equilibrium reaction, it is substrate product inhibited, it is also inhibited by cyanide, which is introduced in the second step, and the biggest issue we found was the problem with the operational stability decay of the enzyme introduced or influenced by this compound here. So this means that if we put too much of this compound to the reaction mixture, the enzyme will lose its activity. In the second step, we also found that the reaction is product inhibited. And in this case, considering that we are dealing in epoxides in water system, there are chemical side reactions which have a serious effect on the outcome of this reaction step. As far as the third step, we found that this step is also product inhibited, and it is inhibited by substrate of the first and the second step. Thus, I think from this you can see already that it's a highly complex reaction system and that we need to see and we need to evaluate what is best to be done. Now, just for as an example, I will show you here So we modeled the entire system, and for the first reaction step, you can see that we could evaluate the validity of the model, and it describes the data well. And what we could also see is that operational stability of enzyme is seriously influenced by the substrate. Thus, what comes up as a solution is a fed batch process in which you add a 1,3-dichloro-2-propanol in order to save the from the damage. So in this case, we could obtain around 100 millimolar concentration of the product. However, so I'm talking about this product here. However, we were not really happy with this. We wanted to go higher. So we thought, okay, what if we leave out the first step of the cascade? What if we start from the second step? We could also use carbohydrate. Considering that this is, in water system, tend to hydrolyze. Thus, in this case, we could go up with the concentration of the product. However, this was still not something we're happy with. It's because all these
worst case scenario, we lost the speaker.
Okay. Yeah. So let's wait a few seconds. See if she can rejoin. Yeah, she's back. Okay. Hello, can you hear us?
Can you hear me?
Yeah, we can hear you. Yeah.
Yes.
Yeah, unfortunately,
Okay.
we lost you during this last slide. Roughly in the middle, it started to break
Previous
off.
one? Okay, so previous slide, you mean?
This
one?
This slide, yeah, when you started to
Okay.
explain those the two last graphics, the fed batch optimizations.
Oh, okay. So the reason why we chose
and we cannot see your pointer in case you were pointing something out.
Okay, I only
now
use the
mouse.
this.
Okay, yeah. So I couldn't always see it. Now I can see it. Okay, carry on.
Okay, thank you. So as far as the choice of the fed batch, we chose it because the effect of this substrate is severe on enzyme activity. And in this case, we could obtain around 100 millimolar concentration of the product. But we were not happy with this, so we considered, let's start from this compound here. Considering that it's an epoxide in water solution, it can be unstable. We thought, okay, let's start with the fed batch process again. And we could obtain a bit higher concentration of the product. But still, we were not happy with it. Considering that this compound here, as well as this compound here, are really cheap, this is a bit, let's say, more expensive, but cheap in comparison to this product here. We wanted to see if we can use the third step to obtain better results. So as in all the cases, we investigated the kinetics and we set up the model, and we found the best conditions by using the modeling approach, the simulations. So we said, okay, let's do the fed batch for the third step. We will add this compound here to the reaction mixture, and then we obtain these data. So by using this approach, we could obtain about a bit more over 100 millimolar concentration of the product. And the conversion was really good, around 97%. Then we isolated the product. The isolated yield was a bit lower because it was a first time isolation, so it wasn't a perfected procedure. But we were able to obtain a nice amount of product, which was found to be very pure and confirmed the structure by NMR analysis. Now, this is as far as the first reaction system is concerned. Now I will go on with the second system I'd like to show you here, and it has been investigated in collaboration with Margit and ASIP in the project Circle H2. So we are studying the carbonyl reductase catalyzed reduction of octanoic acid to octanol, and we are using the same approach. As you can see here, there is a regeneration of coenzyme that is necessary, and there is a region of NADPH, and also regeneration of ATP. So all these enzymes, all these reactions have to be investigated. And what I'd like to really highlight here, it is very important to have a really good analytic system set up. So in this case, it's both GC and HPLC, depending on which system we are talking about. So to start with, we started with the kinetic characterization of reduction of octanoic acid. So this means this reaction here. And in this reaction, so we are selecting the first sort of step of the cascade, we wanted to see and to evaluate the influence of all reaction compounds on the reaction rate or specific enzyme activity. So this is the data that we collected. And what we found later on is that enzyme also catalyzes the reduction of octanol to octanol, which is unwanted side reactions. So in this case, we also evaluated the enzyme activity in this reaction in order to evaluate how strong it is and how it will influence the reaction outcome. So from the presented data, we could estimate the kinetic parameters that are presented here on this slide, and then we were able to set up the kinetic model. So for the model, we are still working on model validation. So this is something that still needs to be carried out. However, the second step, the third step, the coenzyme regeneration, this is something that is going to be done in the frame of a Circle H2 project. So what is also of very much of interest for us is the stability of enzymes. So in this case, we did not evaluate operational stability of carbonyl reductase yet, but we did test the storage stability of the enzyme at different temperatures, and we found that the enzyme can be stored for quite a long time at both temperatures. So I hope in some other webinars or workshops you will hear about new results in the future. Now I would like to say something about techno-economic assessment, which I mentioned earlier. So a part of our work in different projects, including Circle H2, is also estimation of economic part of the process. This means that we need to establish what is the price of the product to be obtained by using the synthetic approach. And this is sort of a methodology that is being used and which ends with a quantitative risk analysis using Monte Carlo simulations. And as a result, you obtain a figure like this one, where you can say that the estimated price of the product is a certain price, which can then be compared to the commercially available products or similar products, just to have an idea if the project is viable to be scaled up, or maybe it is better to stop investigating certain reaction system at a low scale, because as you know, the process development costs a lot, and there are lots of projects that are killed during the way. So early stage analysis and early stage decision making is quite important. Of course, everything I said earlier about the system, about performance, reaction conditions is very important for this estimation as well, together with the downstream processing procedure. So I'd like to conclude on the modeling that even though we work on small scale to begin with, these kind of information can lead to very well-defined models that can be used for a lot of different calculations. Of course, on higher scale, we can see that non-ideality can have an influence on the outcome. However, it is important, even on the low scale, to evaluate what are the side reactions. Is the enzyme being deactivated during the reaction? Are there any solubility issues? Because this will help us later on in process development. So understanding is something that is very important for the process development, and mathematical model can really help us with that. So with this I would like to first of all thank all people that are included in the results I presented today. Of course, I would like to thank funding agency, and I would like to also thank you for your attention and for understanding for the problems with the net. Thank you.
Thank you for this really nice presentation. Luckily, most of it was very easy to understand, and the internet connection held mostly. So it's my pleasure now to moderate some questions. So if you have questions, please raise your hand or put them in the Q&A. In the meantime, since I'm moderating this, I have the privilege of actually asking the first question, because I do have a more methodological question. So on slide 16, where you showed the stability of your enzyme, you talked about the operational stability that you want to evaluate. And I was curious,
Yes.
how are you actually going to look at the operational stability? Because for the storage stability, I've done this myself for enzymes. I guess this is quite straightforward. But how do you look at the operational stability?
So for the operational stability, we usually take samples from the reaction solution, and then
you
use
just
it
cut
in
out.
the
So
assay.
sorry, you just cut out. So you take a sample from the solution, and then you go directly into the assay, or?
Yes, but first we separate and wash it with the buffer to remove the substrate from the reaction solution. So usually it works quite well with the filters, with the cutoff specific for the enzyme.
So you use ultrafiltration.
With some enzymes, exactly. So with some enzyme, it can be problematic because they can be, let's say, more sensitive to this procedure. However, with CAR, we already tried it and it works really well.
Okay, great. Thank you. That makes a lot of sense. Yeah. Okay. Are there any other questions? Margit, maybe?
Yeah, while other people are still thinking, I could ask a question. So, Svetlana, what is the success rate of your methodology when it comes to like multi-step reactions? So, can you compare this to the traditional way of reaction optimization? So, how much can the kinetic modeling speed up the process?
Well, I would say that it's a very successful methodology. We did numerous cascade reactions, and we could really increase the productivity or the concentration even by 100%, or the conversions nearly to 100%, with the systems that were initially, well, almost disregarded as useless, so to speak. So even when you do the screening of enzymes, sometimes if you choose the wrong concentration, you will get no results for activity. So I would say it is very important to know at what concentration points your enzyme will be the most active, so to speak, or at what concentration points the enzyme will be the least inhibited. And considering the complexity of the equations that we have here, I think that simulations really help a lot. 4 steps or even 2, I don't know. Easy to use the simulations and to check out how the system behaves when you want to change the reaction conditions or change the I would highly recommend the modeling approach because it's quite straightforward. It gives, well, if you get some out of your system or not.
Okay,
It's
very
difficult to get the point. It's
we have
again,
some connection problems.
there are some questions.
Yeah,
So
nice. Yeah, we have two questions from the audience. I don't know. Should we? I mean, I think we got most of your answer.
can you hear me?
Yeah, you are back. Yeah.
Yeah, let's try to.
So should I read the questions myself?
No, I was going to read one. Yeah. So Cristiano
Okay.
Cristiano Varone asked, what can be done to minimize the secondary activity of CAR on octanol without going for mutagenesis?
Well, I would say that it is important to set up the reaction rate of the first reaction step so that the concentration of the substrate or substrate for the second reaction is
you cut
well
out.
You cannot hear me?
You cut out in the middle of the answer. So.
Okay, sorry. I would say that reaction conditions needs to be set out in a way to accommodate them to perform the first reaction. When this CAR reaction will be combined with a coenzyme regeneration system, it will be much easier to handle this. So I expect that by using the model, we will be able to solve this. Not entirely, of course, but we had a similar case where there was over-oxidation in the system where we could really obtain 92% conversion in the end, yield, sorry, towards the wanted product. So I expect that we will be able to go there by using the model.
Okay, I hope this answers the question of Cristiano. We have two more questions, anonymous questions. The first one is, can you explain the productivity in comparison to metabolic engineering of microorganisms?
I'm not sure that I understand exactly what is meant by this question, but is it to compare the productivities if a reaction is performed by using enzymes and microorganisms? So if this is the case, then we had something like that as well, where we used purified enzymes, cell-free extract, as well as microbial whole cells with co-expressed genes for the targeted enzyme. And we could use the model built by enzymes, I mean by enzymatic system, to optimize the system in the cells. So I hope this was the answer to this question. So of course, in microorganism, there is more side reactions, which needs to be taken into account. But in our case, in one system we studied, it was really successful.
Okay, and then we have quite a few coming in now. Nice. So the next one is, while modeling, is it fine to assume the parameters value like enzyme adsorption or desorption to the substrate?
I have to admit that I have never used such parameters.
I guess this
to
would
begin
then
with.
be for a solid phase reaction, I guess, if you have to work with adsorption and desorption parameters, right? But I guess what you presented was all in solution or
yes, what I
soluble?
presented is all in the solution, exactly, yes.
Yeah, but
So
have you
every
worked with,
parameter.
have you worked with solid phase enzymatic reactions as well?
Immobilized enzyme, if you mean that, yes, I have. And we measure the kinetics in the same way. So
So
we did not
does
use
does the kinetic of this adsorption desorption then actually become part of your enzyme kinetic in a way, or is it a separate parameter?
I would say that no, it's not a separate parameter. It's an embedded, I would say,
Okay,
in,
so it's
the
a lumped.
parameters we estimate. Yes,
Okay.
apparent values of these parameters.
I hope that answers the question. Yeah. And then Barbara Petschacher is asking, what is your experience? How easy is it to get closed mass balances, especially if you have several steps? If you
Well, that
actually
is our
like
target
complete
that
mass
we want
balance, huh?
to, yes, that is, we have to have a closed mass balance. We have to know where our substrate ends up. That's why we need a really thorough analytic procedures to evaluate all the compounds or to quantify all the compounds in the reaction mixture. So if we don't get a closed mass balance, then we search the reason why, and we usually find some side reaction that is going on, and then we figure out what is the side product.
And how close to 100% do you need to get in order to
Well,
perform the
I
model?
would say that it's always in the range of 90, over 95, 96 and above.
Okay. Yeah.
Usually we don't have such issues.
Makes sense. Okay. Yeah, in the interest of time, and since there are no more questions right now, thank you so much for your talk.
Thank you.
And the answering the questions. If you can think of more questions, feel free to post them. Yeah. So after the next talk, there's another opportunity to ask some questions and have a chat. Yeah. So with that, I'll hand back to Margit to introduce our next speaker.
Yes, I'm very happy to welcome Aditya Kunyapur. He's actually American. He did his bachelor studies at the University of Texas, Austin, followed by a doctoral thesis at the MIT in Boston. And then he did a postdoc at the Harvard Medical School, and right now he's associate professor at the University of Delaware. Aditya is a metabolic engineer and has a passion to use non-standard amino acids. Fun fact, during his PhD, he developed a strain that's really, really very useful for us. There are particular knockouts, and we are using this strain for 10 years now, and we are also recommending it to labs all over the world. So it's really a very nice strain to work with. And the respective checks paper that he published in 2014 has meanwhile 361 citations. I looked it up today. Now, currently he's on sabbatical in Denmark. And then for those who are just around the corner, he is going to visit us in Graz in November. On the 12th of November, he's going to give a talk at our institute. And he's also visiting after us the TU Wien and giving a talk there on November the 13th. But now I'm not taking any more of your time. Please, Aditya, share your slides and go ahead with your presentation. Thank you so much for accepting the offer to speak for us.
Oh, of course. Thank you so much, Margit, for the opportunity to present today, and Nico. I really enjoyed seeing the last presentation as well, since, as you know, I like the carboxylic acid reductase enzyme quite a bit. So that was nice to see. I'm going to go ahead and share my slides, and for the moment, I'm keeping my video on, but I'm just curious, how is the internet connection? Is it smooth so far?
So far,
good.
we can hear and see you. So let's see.
Okay.
But can you share the presentation?
Yes, of course.
Yeah.
And I think maybe out of precaution, I'll turn off my video just to see. Hopefully people won't mind that. As Margit mentioned, I'm an associate professor at the University of Delaware, and currently I'm also at the Technical University of Denmark. So it's really great to have opportunities like this and the upcoming visits to Graz and Vienne that Margit mentioned to really interact with the European biocatalysis and modeling communities. We are an experimental first kind of group, but we are dabbling more into modeling. Let me see also, let me make sure that the video controls are not blocking your view. Okay, great. So today I wanted to tell you a little bit about our modeling efforts, specifically for a new metabolic interaction that we've engineered. And that would be, let's see, I just don't want to block this, but I don't think you all are seeing that anyway. That would be for what we're calling obligate and orthogonal metabolic interactions. Just one quick question to the organizers, since I'm starting around 14:10. Should I try to finish before 14:30 or 14:25?
Well,
Something like that?
since it's being recorded, please take your time to present what you plan to present. We had some, due to the hiccups with the connection, we just lost some time, but don't feel rushed through the slides. Yeah.
Okay, thank you so
Just
much.
be aware that some people might have to drop out at 2:30, but if that's the case for you, please feel free to ask questions in the chat before the Q&A starts if you have to leave early. Everyone will be provided the recording. Yeah, so but I'll let you keep presenting.
Okay, wonderful. Thanks for that clarification, and I'll try to be respectful of everyone's time. So I wanted to motivate this kind of in two ways. Our lab is actually looking at the fact, first, that biotechnologists have engineered microbes to produce all kinds of chemicals and really brought forward capabilities in sense and response to chemicals, as well as breaking down and sequestering wastes or hazardous compounds. And some of these activities of microbes could actually be useful in the environment, especially because many hazardous or waste compounds are actually distributed in the environment rather than collected for centralized bioprocessing. And so for these cases and others, it would be nice if we could do it responsibly to be able to look to strategies to release an engineered microbe. And in the United States, this might be a little bit more palatable or closer to approval than perhaps in Europe. But at the same time, we really need to make sure we do this responsibly, because if we don't control the fate of an engineered microbe, of course, they can divide and spread, and they could cause unintended consequences, including something as simple as, let's say, nitrogen fixation. If it occurs at too high a rate in the wrong location, it could contribute to ecosystem destabilization. And so accordingly, the field of synthetic biology has also focused on using these technological advancements actually to make safeguards for engineered microbes. And so some of these are known as intrinsic biological containment systems. And what they are is they seek to program the genome of an engineered organism to survive only under certain conditions. And there are other important techniques that would be required too that I'm not going to discuss today, where you would need advancements in the stability and fate of the engineered strain and its recombinant DNA, particularly to limit horizontal gene transfer. Now, for those who think about more traditional bioprocesses, and in case these applications may seem a little too futuristic, then I should also mention that the kind of topic that I'm describing today could be useful in existing industrial bioprocesses, such as anaerobic digestion or waste treatment, where more than one microbe or microbial consortium, as I would call it, is used. And here, in particular, tools to control the abundance of just one species within a population uniquely would offer a precise ability to tune reaction kinetics and product distributions. So now to my group, what we seek to do is to use synthetic biology for both angles here, like the creation of new microbial capabilities as well as safeguards, therefore expanding what molecules cells can make and also where cells can make them safely. And we try to do this using a combination of approaches across metabolic engineering, genome engineering, and chemical biology. So in the first 6 years of our lab, we've focused on enabling these capabilities and safeguards by creating and harnessing non-standard amino acids, or NSAs. So we're programming microbes to make and use these NSAs, and NSA is really just an amino acid where this R group, or the star in this case, is any structure that's not part of the 20 standard amino acids translated by the ribosome. And so the incorporation of non-standard amino acids can be enabled by these approaches called genetic code expansion. They've led to a lot of different applied technologies, some of which you can see here. Typically, someone is chemically synthesizing and adding this NSA, but increasingly, our lab and others are trying to develop cascades or metabolically engineered pathways to make the non-standard amino acid. And I would say that what we're trying to do, especially, which will be one example will be highlighted in this talk, is we're trying to show where if a non-standard amino acid is biosynthesized, that can enable something new. So going back to the idea of the biocontainment for potential environmental release or for control in a consortium, pioneering work from my postdoc advisor, George Church, and from a former Church Lab alumnus, Farron Isaacs at Harvard and Yale, showed in 2015 how microbes could be engineered to rely on a non-standard amino acid. In this case, this is a biphenylalanine. But in any case, you would pick a structure that isn't one of the standard 20, and you would engineer essential enzymes in the cell to either depend on the non-standard amino acid for full-length translation or for proper folding, or both. And that way, if you have the non-standard amino acid present in the media, the cells have their essential enzyme work and they grow. And then in the absence of the non-standard amino acid, even if there's a mutation, you would expect there to not be function, and so there shouldn't be growth. So this technology, at least in the case of one engineered strain, worked at controlling the E. coli strain such that it required BIPA, or this biphenylalanine, for growth. Even up to growing 1 trillion cells, there was still no escape from this reliance when culturing and then washing away the BIPA and plating on media that wouldn't contain BIPA. So we have systems where we can engineer microbes to tightly rely on a non-standard amino acid. However, there's some limitations of different kinds for trying to imagine how this would translate to an environmental application. Because by design, this non-standard amino acid dependent microbe, otherwise known as a synthetic auxotroph, well, it's going to die if it doesn't have its non-standard amino acid. And of course, you want that level of control. But for many applications, be it a plant probiotic or bioremediation, you may actually need the microbe to survive for longer time periods, but in a defined spatial location within the environment. And so we thought about this problem, and we thought maybe biosynthesis could offer a way of autonomously supporting the dependent microbe. And so as a proof of concept, my student Mandy Forty investigated whether we could design a symbiosis. This would be where a second organism, which for right now is a microbe, but we can imagine other kinds of organisms like a plant. I'll just stick to microbes for this purpose of this story, and that's all we've published so far. You could imagine that one microbe could produce the non-standard amino acid that another microbe relies on for its survival. So here, this is a cartoon depicting a blue microbe that relies on a non-standard amino acid if it's added, in this case, depicting a test tube consortium of other microbes, it won't survive, but in the presence of a yellow microbe engineered to produce what the blue microbe needs, now you have a permissive state that allows the blue microbe to survive. And so we call this an orthogonal obligate commensalism. Orthogonal because it should be exclusive, such that this chemistry is really just one way between yellow and blue, and the other microbes are not really making it. In many cases, they may be degrading it, which I'll briefly touch on. But they are not able to support blue without yellow there. And obligate because it's required for the survival. So, and commensalism because it's a one-way, not rather than a two-way dependence that's not harming nor the yellow microbe. So this is a kind of a transition from a chemical dependent strategy into now a biological or ecological dependence. And this is the experimental system that we took to modeling. But now, before I dive right into the model, I still need to tell you a little bit more about the experimental system. So in short, this proof of concept that we were lucky to publish in May of this year took 2 E. coli strains and engineered one as the producer to biosynthesize a non-standard amino acid from endogenous metabolites that's shown here, O-methyltyrosine from tyrosine through a simple methylation. And then there's a utilizer microbe. And this actually took a long time to engineer well. It needs to take then the O-methyl tyrosine, now depicted as this pink pentagon, and place it into essential genes. And then, have that tight dependence. We also weren't initially sure, there are many different non-standard amino acids, so we tried to enumerate the criteria that we would use to pick a non-standard amino acid. And of course, for an environmental application, you wouldn't want it to be present widely in nature. You wouldn't want it to be very reactive either. I'm going to talk a little bit about 3 and 4 and why they're relevant, especially to modeling. This kind of simple biosynthesis is very important, as this next slide will kind of show, because you need, in this system, one microbe is independent of the other. And so the dependent microbe, the utilizer, needs sufficient non-standard amino acid to be built up quickly enough for it to then grow. But by that time, you have biomass formation of your producer microbe. So we looked very closely at the rates of non-standard amino acid biosynthesis under different media conditions. Here you could see plotted the concentration and culture time. And so it's really during exponential growth in a monoculture batch shake flask where you could see that it takes 24 hours to get near millimolar concentrations, but this is not really a metabolic engineering project so much as how much amino acid do you need for the other microbe. And so that's where it's kind of interesting to look at this other criterion of the site-specific incorporation, and specifically at the affinity, which is measuring how much is the half maximal concentration of non-standard amino acid required to translate proteins. And so we're very lucky that the system that we picked only required 0.03 millimolar. And so while this is of a fluorescent protein reporter that we can easily measure, we imagined it would still be the same order of magnitude when we eventually make the cell depend on it. And so you could kind of do some rough heuristic here, like 0.03 millimolar. We get there pretty quickly. So, the second microbe its outgrowth is delayed, but not by too long. So as I'm about to get to the modeling kinetics, just want to show you that the system does work. And how we're kind of evaluating things when we're also comparing with our model. If we have just the blue microbe that's engineered for reliance on O-methyl tyrosine, and we plate on media that is selective for just the blue microbe because it has a different antibiotic resistance, and it also permits growth of any cells that would have been in the culture because it has the non-standard amino acid on the plate, we see that the blue microbe wasn't able to survive on its own. And that's why this plate is empty. It's not able to survive when you provide a non-producer E. coli, but it can survive when you provide the producer E. coli as a co-culture. And excitingly, this kind of concept also holds up in the synthetic microbial consortium, just a small, simple one of antibiotic sensitive microbes derived from soil from a synthetic maize root consortium. And that's where we also see that still the blue microbe relies on yellow. Okay, but now to the focus of this presentation, our ongoing efforts to mathematically model this system and related systems. And this was really work led by Michaela Jones, who's now a postdoc at MIT. So the rationale for this modeling effort is to predict the abundance of each strain, to determine more broadly how to optimize the current system, and then also to quantitatively predict how other interactions and other systems would perform. And so as I kind of highlighted earlier, we know that the productivity, or we had at least an intuition that the productivity of the NSA biosynthesis and the affinity were going to be important parameters. There's a lot of work well established in the area of cell growth kinetics. The modeling of bacterial growth is well described by Monod growth kinetics. And we can also use, so here are some of the Monod growth kinetics for the producer microbe. We can also use mass action kinetics to prepare ODEs for the key metabolites. This is an ODE actually of the biomass of the producer, but here are some metabolites in gray. We have our non-NSA limiting nutrient. You can think of this as like the carbon source, probably glucose. And then here is the NSA concentration, which is related to the biomass of the producer through a yield coefficient and its growth rate. Now, since the utilizer requires both the limiting nutrient and the NSA, we opted to use a published adaptation of Monod growth kinetics for double substrate limitations. And that's when you have a dependence on two substrates. And you can kind of see that here with these inequalities on the right, where you could be limited by either the availability of the limiting nutrient or the NSA, depending on how much of each there are relative to the half maximal constants. And so this is innovative in its application to this system, but it was published in 1978, given that sometimes there can be regimes just naturally where maybe a carbon and a nitrogen source are provided in similar quantities, and therefore you might have two substrates that are affecting growth. So here you can just see a more complete list of our defined variables and their units. And I'd like to highlight that for all the simulation data that I'm going to present, the biomass is in units of milligrams per liter, whereas most of our experimental measurements are in optical density or colony forming units, and work is ongoing to estimate the conversion between those units for E. coli strains. So now, to model development, one of the first steps is parameterization, and we do this using a mix of literature and experiments. And again, I apologize to anyone who is, routinely an expert in modeling, because this may seem very basic. But for us, this was kind of a newer space. And so what we thought, especially with this system, was that it would be important to derive the parameters for our engineered strain and for everything about the NSAs through experiments. So here you can see an example of how we titrated the concentration of the non-standard amino acid and can see how it affects growth rate. So here we've plotted now the growth rate, and we've now fit a nonlinear regression. And from this, you can obtain both the max specific growth rate of the utilizing microbe, and you can determine the half maximal constant, the affinity. And we did also borrow some parameters from the literature. For example, the affinity of E. coli for glucose, or that's within a pretty well-documented range. And we don't expect our system to really change that too much. So I'm going to run through then what are some controls we applied. We next essentially tested a lot of control conditions that we had also experimentally verified, just to kind of see, ballpark, are we getting the right kind of abundances? So typically what we're doing then in these plots, we'll have a plot where we're tracking the nutrient concentrations, the NSA concentration, and the biomass of each species. And then you'll see on the right a more focused graph, zoomed in, much different y-axis here, of growth of the utilizer. And there are many regimes where the utilizer struggles to grow. And some of these are deliberate. So these controls, the utilizer is predicted to be excluded, but when it's by itself or with a non-producer, and the non-producer is able to grow. So our simulations on the right are matching with these qualitative plate images. Then we have the more interesting controls. You've got the two strains and you're providing the permissive non-standard amino acid. This is exogenously supplied. So this is now kind of a best case scenario for the utilizer, how it just has to compete with the yellow microbe, and it does exhibit a little bit worse fitness. So therefore you see the producer is in whatever milligram per liter units, somewhere above 300, and the utilizer is getting close to 100. And so they're comparable in order of magnitude, and that's actually what we saw experimentally. And then the case, though, where there is no exogenous supplementation of the non-standard amino acid, so blue has to wait for yellow to make it and for it to slowly build up, well, then you can see what happens. And even just visually between these two plate images, it's qualitative, but when we did more careful quantitative analysis, we saw experimentally we're about 3 orders of magnitude lower when we go to this case of blue relative to what it would be here. And actually, when you compare these simulated results, we're about 2 orders of magnitude lower. So much of our modeling then was oriented around trying to improve representation of the utilizer. The other thing I'll just say is that we did also, because we could select for each microbe, in our co-cultures, we did this extensive time course analysis, where each one of these dots represents plating in triplicate CFU. And you can see the CFU values plotted on the left y-axis for both the producer, where we see excellent agreement over 24 hours, and the utilizer, where the agreement is good, but it's not, it could improve. And of course, on our y-axis on the right are milligrams per liter, so there's a little bit to iron out there. But we were happy to see this level of qualitative prediction. So now I'm going to get into 4 quick things that the model could be useful for, and then I'll basically wrap up my talk. The first, in this ecological kind of scenario, is essentially trying to look at regimes of coexistence or exclusion. So Michaela had the idea to gain essentially this insight into how the system could behave rather than just how it does behave by creating something analogous to what many engineers would view as like a phase diagram. And so the axes here are those 2 key parameters, the affinity for the non-standard amino acid and the product yield. And you can see now ecological regimes ranging from exclusion, where the utilizer microbe will perish, to survival, where it can kind of manage a non-zero but abundance, but it doesn't increase relative to its inoculum, and growth, where it actually does go above its inoculum, sometimes only to go down later. And you can also see where our co-culture sits. So this gives us some sense of what we might need to do to improve our system, and how much of an impact that might make, though more on that is coming. By the way, all of this is unpublished data, and so if you have any input or suggestions on what else we could do with this model, we would love to hear it. One of the things that we did do in our Nature Microbiology paper is we did actually slow the growth rate of the producer strain by making it depend on a different non-standard amino acid. And we saw that that was able to increase the abundance of the utilizer. Our simulations indicate that such a thing would be expected. So, and this is important for considering any set of different hosts. Here, what you see are simulated lines that would be the boundaries between the survival and growth regimes. And so where we started was on this pink line, and where we ended with a certain case was on this orange line. And what this orange case shows you, just to explain it sort of in different terms, is you could theoretically achieve coexistence, which is to the right of this line, under more combinations now of NSA yield and affinity. And we, especially for environmental applications, would be thinking about moving the system into other kinds of hosts that are more environmentally relevant. So this is important considerations for us. Also, in our paper experimentally, we tried to show that mutualism could increase abundance. And we made a simple producer modification that made it a histidine auxotroph, so that it relied on the other strain too. And here you can just kind of see that our modeling framework is such that we can very simply impose a double substrate limitation on the producer too, and start to play around with different cases where maybe it would make a second non-standard amino acid, and then they would mutually rely on each other, and we could look and see what the growth could be like in that case. And so then the fourth and last thought that I have on how the model can be useful to our experimentation is for parameter sensitivity analysis. You can use, we're still early in this stage, but you can see the impact of different parameters. And as long as your parameter ranges are realistic, then this can really help guide what kind of optimization to do using engineering. So lastly, I think it would be appropriate to conclude with like some limitations. I've already touched on the milligram per liter to CFU kind of discrepancy, but I think to those who are well familiar with metabolic modeling, they would know that there are many alternative strategies. I guess, well, what I should say is that actually a good example to motivate this is if we think about the parameter sensitivity analysis again, when we've developed this model, each parameter is independent. However, we know experimentally that if we engineer the producer to make more O-methyltyrosine, thereby increasing its productivity, that will carry usually a detrimental impact on its maximum specific growth rate. And experimentally, this isn't a big deal because we can just remeasure the specific growth rate. But computationally, what one could do if you integrated a genome scale metabolic model is actually then you would be able to understand how the flux towards the non-standard amino acid under different conditions would be predicted to interact with the specific growth rate. And that kind of modeling is very feasible. And in the long run, it may make sense. But in the near term, we aren't pursuing it yet. You could see even from this review from Bernard Paulson's group that there's interest in applications to synthetic ecology there. Okay, so then take a moment to summarize. So what we designed was a new metabolic interaction that was both exclusive and required for an engineered microbe. And we hope it can facilitate the control of the abundance of the engineered microbe in a more autonomous manner within mixed microbial populations. And then to better understand and improve the system, we turned to mathematical modeling. And we're happy to see so far that our model qualitatively agrees with our experimental observations, and it's really helping guide our next steps. So with that, I'd like to acknowledge the students who performed the work and the funding agencies who supported it. I'd like to thank again the ACIB hosts for inviting me to give this talk, and thank the audience for listening. And if there's any time, I'll take any questions that people may have.
Thank you, Aditya. Great talk. Thank you. So to the audience, please feel free to raise your hand or post questions. I want to thank both you and Svetlana for showing the potential of thorough modeling even early on during development to serve as a guide for further development. This is actually quite a nice lesson here. Yeah, models can and should be used quite early on, right? I was curious. So, what is holding you back from including the metabolic modeling into your full model? If I may ask.
Sure. I think some of it may have to do with the idea that for the utilizer, the dependence on the non-standard amino acid is not a kind of a straightforward metabolic step. It's that it gets involved in the process of protein translation and in a highly engineered way. So at least for modeling the utilizer, and the utilizer is also not engineered to biosynthesize anything unless it's in the mutualism cases. We thought probably the monode growth OD approach would better capture that empirically. But definitely for the producer, we could, then it's just a matter of experience. I've used at times Cobra Pi or some of the other packages for the constraint-based optimization, but not like it's the back of my hand, let's say that.
Okay, yeah, that makes sense. And the monokinetic approach seems to work quite nicely. Yeah, so makes sense. We do have a question from an anonymous attendee. So they are asking, when I am modeling, I used to take kinetic values for different enzymes from different literature. In that case, how will I validate the model computationally? I don't do experimental work. And can you give me a suggestion of what I should be careful of when doing that?
Well, that's a great question, and I really appreciate that our computational research community is trying to be thoughtful in that way. And so maybe I can try to answer this question. I think I wonder in the case of this, like the cascade approach, probably the Michaelis-Menten kinetics are better determined, but we saw from the last talk that, even how they're stored and how long they're used can impact the activity and rates. But trying not to think too much of the last presentation, rather my own, well, yeah, I think the actual Km in a cellular context of an enzyme could depend first on how efficiently the transport and uptake of the substrate may be, particularly if it's being provided exogenously or made by another organism, right? So in our case, I don't know how much it really affects O-methyltyrosine, but we have certainly seen cases from our other work. And I think a well-known example, maybe in the biocatalysis community, is for plastics, something like terephthalic acid for PET degradation. Many times the intracellular concentration of TPA is quite different than what's extracellular, and it's limited in many cases by the transporter. And so then, even if you had the KM, you would just need to make sure to also have information about transport. And I think our last speaker also referred to byproduct reactions and the hypothetical question about metabolic engineering case. So in our case, one thing I didn't touch on is that the O-methyltyrosine in the larger synthetic community is actually degraded slowly by other microbes. But this is actually a good thing. Otherwise, the dependent microbe would become independent of the producer, and you'd have the non-standard amino acid pollute and build up in the environment. So it's kind of good that the non-standard amino acid is degraded in the environment.
Yeah, that's a good point. Yeah. Okay. Are there any other questions?
Yeah, I would have one actually. So we also tried to work with growth assays, and then I realized life is really strong in finding ways to also survive without the compounds that you want them to rely on. So did you have any struggles with false positives? Because in my experience, it's rather difficult to really do this kind of selection.
Yeah, absolutely. Let's see if I can go. I'm not, for the sake of simplicity in this talk, I didn't include any of our escape rate data. But the blue strain that we engineered does still exhibit an escape that's observable from biocontainment. I think probably many of us in the context of directed evolution have maybe tried using an antibiotic resistance marker as a selectable marker and know that sometimes on the order of 1 in 10 to the 7 or 10 to the 8 will be something that we'll call a cheater. So here, I just mentioned that for reference, the blue microbe that we finally report in our paper, after 14 days of incubation of a plate in the absence of the non-standard amino acid, we see 1 in 10 to the 9. So we have a 1 in 10 to the, well, it's a 10 to the minus 9 type of escape rate from containment. And so you could argue that that's good for the purposes of the containment field in the literature to demonstrate a concept that's more ecological, but it certainly isn't good enough for environmental release, where, at what scale would you, for agriculture, for example, it would probably be a lot of microbes. However, for human health applications, the US NIH has a 10 to the 8 kind of reference point, escapees per CFU metric. And that's because I imagine for probiotics, people are administering a much smaller microbial dose. But you'd like to be many orders of magnitude below that to avoid stochasticity and the possibility of escape.
That makes a lot of sense. Yeah, life finds a way, right?
Yes, unfortunately.
As Jeff Goldblum's character in
Yes.
Jurassic Park said. Yeah, I would also be worried about that. I think the escape rate would have to be a lot lower. All right, it doesn't look like we have any more questions. But yeah, feel free to reach out to us or any of the speakers if you can think of anything. The recording will be provided to all the registered users. And yeah, I want to thank